Welcome students. This module is for program Bachelor of Science, first year, Botany, semester 2. Course code is BOG 102. Course title is Coastal and Mangrove Ecology. Title of this unit is Flora and Fauna of Mangroves and Associated Environment. And in this module, we are going to learn bacteria, fungi, and actinomycetes which are found in mangroves, collection, preservation, and identification techniques of these. Myself, Ashish Prabhugamkar from DCT's Tempe College of Arts and Science, Goa. In this module outlines collection, preservation, and identification techniques used for bacteria, fungi, and actinomycetes found in mangrove ecosystem. At the end of this module, we should be able to explain about the methods of collection, preservation, and identification of bacteria, fungi, and actinomycetes associated with mangrove ecosystems. Mangrove ecosystems are rich in biodiversity. True mangrove flora consists of only 35 species which are belonging to 16 genera and 13 families. So biodiversity in mangrove is largely represented by other groups of organisms. Other floral species such as actinomycetes, algae, fungi, seagrass, bacteria which are found in mangrove remain less understood and less explored till date. Total floral species reported from mangroves are 420 species and faunal species 1442 species. You can imagine the extent to which the species remain unexplored. Floral species reported to exist in mangroves in India according to Kathiresan et al. In 2000 is bacteria 36 species, fungi only 84 species, algae 206 species, seagrass 11 species, mangroves 35 species, mangrove associated flora 48 species. We can only say that mangroves and true mangroves and mangrove associated flora are well documented, others are yet to be documented properly. So let us look at what are the methods of collection and isolation and preservation of these organisms. First, bacteria. Bacteria are, as you all know, are microscopic unicellular prokaryotic organisms. They are extremely tiny, and when cultured on agar, bacteria grow as a slimy colony that contains many individuals. These are main three forms of bacteria: cocci, bacilli, and spirilla. For collection of bacteria, we need to collect from mangrove sample either water sample or sediment sample. Water sample should be collected in airtight, sterile bottle. Should not be getting contaminated from other sample. Sediments are collected in either sterile petri dishes or sterile polythene bags which are zip sealed. Samples collected for isolating bacteria should be examined as soon as possible after collection, and if the samples need to be transported or kept for a long, they should be maintained at 4 to 10 degree Celsius, not the freezing temperature and not above 10 degree temperature. In laboratory, samples are generally diluted and they are then spread on the media in a sterile petri plate to get bacterial cultures. Water samples are first filtered through sterile Whatman number 1 paper to remove unwanted debris, and then filtrate is directly used as an inoculum, or further the filtrate can be further filtered to the 0.22 micrometer filter paper which will collect the bacterial cells on the surface of the filter paper, and such filter paper can be again used for inoculating the petri dishes. The sediments are first dissolved in the 50% seawater solution in 100 ml of water, and a serial dilution is carried out. And a serial dilution, 1 ml is taken from the 100 ml and put in 9 ml of water, then again another 1 ml. So dilution of 0.1 ml concentration of the original concentration is taken for plating on a agar plate based on standardization of the procedure. Identification of bacteria. Gram staining is one of the important techniques. There are several biochemical techniques available in identification of bacteria. Gram staining, the two dyes, crystal violet and safranine, are used. Crystal violet stains the peptidoglycan layer on the bacterial cell wall. So the cells appear violet. They are gram positive, and those which are not stained are counter stained using safranine, and then they appear pink, so they are gram negative. This is the procedure for gram staining method. Preservation of bacteria. The sequences generated for identification of bacteria through PCR and sequencer can be deposited in gene bank. Microbial cultures are maintained for long duration in active state by frequent subculturing on the agar plates and agar slants. Under suspended metabolic conditions, bacterial cultures can be kept for years together, that is cryopreservation and deep freezing. Cryopreservation is -265 degrees Celsius temperature and deep freezing at -80 temperature. These cultures can be stored almost permanently and can be used for further bioprospecting studies. Coming to fungi, fungi are eukaryotic, spore-bearing, a chlorophyllous, heterotrophic organism. The main thallus is the mycelial structures. They generally reproduce sexually also and asexually. They are usually filamentous mycelial states, except yeast, which is unicellular. Marine fungi play a very significant role in degradation of litter of mangrove ecosystem. So lignicolous fungi play a very important role, and 450 species of marine fungi are reported. 150 species are reported from mangroves. 72 species are reported from India. Collection and techniques for fungi. Substrates which are collected for identification of mangroves will be dead and living plant material. Fungi exist as a degraders of litter, as a major role they play in environment. So they are also found in humus, they are also found in soil. Interestingly, they are also found as endophytes associated with the living healthy plant tissue. The animal dropping, coprophilous fungi is a group which is found with most of the animal dropping. So all kind of samples can be collected for isolation of fungi from mangrove ecosystem. First samples are directly examined under stereo microscope for presence of ascocarp, basidiocarp and pycnidia, and such spores can be transferred on agar medium, petri plate, sterile petri plate with agar medium to get the cultures. Wood baiting technique is another method wherein we artificially introduce samples from the substrate from outside to be colonized by fungi and then sample them. There are several other methods which for isolation of fungi which I have listed here. Identification of fungi is generally done based on morphological method, that is ascospores, basidiospores and conidia, their shape and sizes. If the fruiting bodies are not developed, only if fungus is at mycelial state, it can be incubated for fruiting bodies to be developed. Preservation of fungi. The same techniques as described in bacterial samples can be used here for preservation, that is cryopreservation and active culture which can be live cultures can also be preserved. Additionally, fungi can be maintained also as a herbarium, which can be deposited in national fungal repositories, which maintain the herbarium for long duration. Our last topic is actinomycetes. Actinomycetes are unicellular gram-positive bacteria. They are fungi-like organism, but they come under bacteria. Recently, they are termed as actinobacteria also. Members of this group are widely distributed in nature, and they can be found in variety of habitat, just like fungi. Actinomycetes are unique microorganisms having both characters of bacteria and fungi. The sediments should be collected with the Peterson grab, and then they should be air dried before isolation of actinomycetes. Isolation is done by serial dilution method, as we have discussed in bacteria. Identification of actinomycetes is done based on the different cell structures and which are of the colonies which are grown on agar plate. Few major groups and its characters are listed here. Streptomyces, we have discrete aerial mycelium, whereas Micromonospora have well developed branch septate mycelium, but aerial mycelium is only occasionally found. Nocardia have rudimentary and extensively branched vegetative hyphae. Cultural and physiological characters can also be studied for identification of actinomycete. Preservation of actinomycete is also similar as bacteria and fungi. These are my references. Thank you.